It's Mr. Maxwell for Pingree AP Biology, and this is for my Period 5 class. This is the list of things that you guys wanted to review. Since we're not going to have, because I can't be in class today, I'm going to do this little screencast for you. And so the first thing you guys said you want to review is the Hardy-Weinberg homework, and I'm going to suggest that what you do is you guys go over that together in class, and if you have any questions, we can deal with that the next time we have class, which should be tomorrow. So I'm going to skip down here to gradualism and punctuated equilibrium. So here's a graphic that shows gradualism. Darwin's theory was that species change gradually over long periods of time, and they change from one form into another with various intermediate stages called transitional forms in between, and that it takes a long time to go from one thing to another. If you actually look in the fossil record, though, what you see is you see long periods of time where you'll have one form, and then in the very next strata, perhaps, you'll see another form. And so this is a problem for Darwin's theory, and Stephen Jay Gould and others came up with this idea of punctuated equilibrium, which states that species generally stay the same and then will go through rapid periods of evolution followed by periods of stasis. And so punctuated equilibrium says that there's an equilibrium that's punctuated by rapid periods of change. And so that's basically the two different models of evolution. It has to do with the rate of change that happens in species over time. So you guys also, if you look at the fossil record, you can take and look at how things have evolved, different transitional forms over time. And so here is a land animal, and then here are our modern whales. And if you want to, you can line these transitional forms up that kind of show a progression from one to the other. Now, take a note that some of these species were, coexist at the same time. The thick line here means that they coexisted at the same time. And so it's not as if this one evolved through all these steps into this one, but that these are forms that existed at different points in history. But what you find in the fossil record really does seem to match a lot more of how punctuated equilibrium would show. Now, it's entirely possible that there are other intermediate forms in the fossil record, but we just haven't found them because we haven't looked for them. Okay, and in case you get the idea that somehow evolution is directional, that the animals were trying to make whales, it's really not that. And this, I think, graphic of horse evolution shows that real well, because you see that there's a route that you go to get to modern horses, but that there are lots of branches and dead ends along the way. So evolution is not goal-directed. Organisms are not trying to make some better organism in some way. All they're doing is doing adaptation to local conditions. Okay, so the next thing is coevolution, and I'm going to talk about the red queen at the same time because that has to do with coevolution. And coevolution is basically that changes in one organism drive changes in another. And in class, I probably showed you the hoverfly, and it has the longest proboscis in the animal kingdom, and its proboscis fits this flower. So the changes would be the longer the proboscis, the longer the deeper the nectary, the longer the proboscis, and so they evolve together. Coevolution happens a lot in mutualistic species, which are one organism benefits from the other, and this would be no different. This is a mutualism here. Parasitism often also leads to coevolution, and here we see that we have parasites and hosts when a particular allele is infrequent and the parasite has to uh, deal with that, it also evolves. So if you think about the host and a parasite, one evolving resistance to the other, that's going to affect uh, population levels of the other. And so what we get is a coevolutionary arms race where the faster one organism gets, for example, the faster the other organism has to get. And you get locked into this coevolutionary arms race where the faster you go, the faster the other organism has to go. And so the Red Queen hypothesis basically is the idea from Alice in Wonderland that you have to run just as fast as you possibly can. In other words, you have to evolve just as fast as you possibly can in order to stay up uh, with this current situation. You have to go as fast as you can just to stay in place. Okay, the next thing we need to talk about is how gene duplications are important to evolutionary novelty. And um, basically, if you think about it, um, if you only have one copy of a gene, that gene has to perform its function. If you have two copies of a gene, one of them can perform the regular function, and then the other one is free to change and evolve. So I'm going to come back to that. We'll talk about the Hox genes. We'll talk about homologous structures next. And so homologous structures are different from analogous structures in this way. Wings are analogous structures. The wing of an insect is part of basically the exoskeleton, whereas the wing of a bird is basically this would be essentially a finger, and then this is analogous. This is the same thing as your forearm, and this is the bone that goes into your shoulder here. In the pterodactyl, the pterodon, you see it's a long finger that gets made into the wing. And of course, in the bat, you have a wing that's made out of the hand, basically. Um, so these are analogous structures. Um, a homologous structure, on the other hand, has the same underlying structure, but not necessarily the same function. So um, the arm of a human, the whole leg of a cat, this would be the flipper of a whale, and the wing of a bat are all homologous because you have this bone, these two bones, there are the two bones, these two bones, the wrist bones here, and then the digits. Okay, and everybody has the same underlying structure, even though these four things are used for totally different purposes. Okay, now if we're talking about genes, uh, genes are homologous, can be homologous as well. We call genes that are the same gene, but in different organisms, we call them orthologs. Because, for example, HOXA1 and HOXA1 in humans and in mice, those are orthologs. But genes can also get duplicated within an organism. And so, for example, this would be humans, and this would be, for example, HOXA1 and HOXA2. And because they are in the same organism, even though they're duplicated genes, we call them paralogous. Okay, so we've talked about homologous structures in genes, differentiated them from analogous structures. An analogous gene would be something that does the same function but has a different underlying sequence. We've explained, so let's explain the function of the Hox genes. And the Hox genes basically are master switches that turn on different body parts and different segments of organisms. And what they want, and here are the Hox genes in each of a bunch of different groups of organisms, and where you see those genes expressed, and they control what's a what whether you put an eye in a particular location or a leg in a particular location. And basically, what these genes are is they're involved uh, in repressing and activating transcription. And so you can imagine if you delete a particular activator, you're not going to turn something on in a particular space where you need to turn it on, um, or whatever. All right, so that's the real quick version of this that I'm going to stop and upload for you so that you have something to look at tonight.